Hello, my name is Lucas Kogan. I'm an MD graduate from the Medical University of Lublin and current residency candidate. And I'm shadowing Dr. Bauer here at Harrison Hospital in Bremerton, Washington, for a clinical observership in internal medicine. And I've been tasked today with giving a talk and presentation on the ins and outs of coronavirus 2019, or COVID 2019, also known as SARS-CoV2. So without further ado, I'd like to begin. We're going to start with going over the basics here. This is going to be a more in-depth look into the coronavirus that's causing all this pandemic and spread out there. And I firmly believe in order to truly develop a strategy to overcome this infection and affliction is to dive deeper into the inner workings of how this virus operates and the origins. So I apologize if this stuff is going to seem a bit more in-depth and may put some of you to sleep. I would hope not. I'm going to try and keep it fun and interesting and clinically relevant. Again, this is more geared towards clinicians, but I'm going to try and explain things in the sense that the public can also digest as well. I don't claim, first of all, to be the foremost expert or a person researching this virus currently, nor do I have any virologist credentials for years and years and years. I just have a general interest in promoting overall healthcare and well-being to the average average human being in the world right now. So I hope that by the end of this presentation I'm able to impart some more information to you, and that you can disseminate proper information to your peers, friends, colleagues. That's my hopes that we gain from this. And if I haven't taught you anything new, I apologize, and in which case feel free to turn off the video at any point. But let's get started. So, points we're going to cover are: what is COVID-19, SARS-CoV2, definition, description, origin, a brief history, snapshot of viral replication process, pathogenesis, including symptomology and transmission. So we're also going to touch base on what exactly a quote-unquote super spreader is, and we're going to go over this S and L type strains and what's the difference, if there is any, and what the impact is on the virus and what we should tell people in the public out there. I'll give you a hint, not a whole lot, but more on that later. Anyways, flu versus coronavirus, we're going to also compare those similarities and dissimilarities, how they stack up, and what, more importantly, to watch out for as symptomology goes. And also, given the limited number of tests available, this is going to help aid our clinical deductive reasoning. In peds and obstetric and gynecology, we're also going to go over the current statistics thus far and special precautions being implemented in these populations or subgroups of infection. And we're going to go over the guidelines as per the current CDC requirement or recommendations on post-exposure and proper personal protective equipment usage. So testing, and that's going to be a major topic of discussion as well to come. Treatment precautions and summary guidelines going forward. CDC recommendations as that all pertains. So, what is COVID? Description, definition, history, snapshot of the replication, pathogenesis, signs, symptoms, transmission. Coronavirus, from the order of Virulence, family Coronavirinae, subfamily Orthocoronavirinae, 4 groups: alpha, beta, gamma, and delta. And what we're going to do is discuss the coronavirus infection of 2019, also referred to as severe acute respiratory syndrome, coronavirus 2, is a new variant mutation of the coronaviruses that have been identified in the early 1930s from poultry populations, and then later on in other subspecies or animal species by the 1960s. So it's a virus that's been around for quite some time, possibly even longer. So it's not like it's coming from the reaches unexplored. We have discovered this virus, but we do not know a whole lot about it yet. My hopes is by the end of this discussion, everyone out there listening to this or watching this will have a general sense, a general idea as to why there's such mystery involved with these viruses and currently what we do know about them. So, and it's been given its name due to its crown-like appearance and the corona and spikes to its glycoprotein, which we're going to talk all about that in the slides to come. COVID-19 is the most recent virus causing widespread infection of humans to date. Duh, we already know this. So what's more important? It's a zoonotic infection, meaning it's arisen from the animal populations we discussed, or that we've briefly touched or reference to. The animals of concern that are hypothesized to have given rise to this strain are bats, and it's been mentioned that 2 species of snakes have also been given rise to this virus, although that's more information and research into that needs to be discovered. So as of right now, bats are the primary animal of interest for this strain. So ordinarily, they're not meant for humans to be infected with, although, and for example, the bats that have been implemented of having the closest strain related, they have completely asymptomatic, non-pathogenic forms and are living symbiotically with the virus in their systems. So, very interesting topic or point of research going forward. So a bit more on the history. Currently we understand little about the coronaviruses. There's only a handful that we've discovered out circulating in wildlife and that have been fully sequenced for comparative phylogenetic analysis to trace their evolutionary origin. And the phylogenetic analysis is pertaining to essentially the family tree of where they came from. And I've highlighted a few of these grandfather type strains, one of them being the PDEV, porcine diarrhea epidemic coronavirus, and the porcine respiratory coronavirus, both of which are shown to be gene mutations of the more virulent and deadly, so to speak, virus of the TGEV, which is the transmissible gastroenteritis coronavirus. So both of those strains are the grandfathers of the grandfather strains, so to speak, to kind of keep it in a general sense. So that covers the general history of where these viruses, or large information we've gotten from the animal populations isolated from those 3 strains, and of which it's theorized SARS coronavirus has been driven from. Okay. And the ability for these viruses to infect other hosts and human hosts and other animal hosts on such a frequent basis is the ability for them to remain for longer periods than normal in the host that they're currently infecting. And when they're allowed to be in that host for longer periods, they undergo more recombination and more mutations that aid the recombination and are able to pick up other virulent factors from neighboring viruses surrounding as the quasi-RNA species. And so that's one factor influencing or describing how these viruses have come to become such a issue, posing a threat to humans. So historically, there are 7 coronaviruses known to infect humans, 4 of which are linked to causing symptoms of the common cold. Those are 229E, O4, OC43, NL63, and HUK1. And it's referenced that approximately 2% of the global population are asymptomatic carriers of these strains that have been found in a statistical analysis. So, and these viruses tend to contribute to 20%. They contribute 20% roughly of cold type outbreaks during the late winter, early spring periods, along with the rhinoviruses as being the predominant viruses causing cold symptomology. So that's just the 4 of the 7. Now, 3 of the 7 are the ones that we reference to the heavy hitters, so to speak. We've got SARS coronavirus, responsible for the 2002-2003 outbreak, and the closest related virus strain to COVID, 2019, and MERS, which is Middle East Respiratory Syndrome coronavirus, that was identified in 2012, and of which we'll discuss the relation to the current SARS coronavirus 2, which is the main star of our discussion today. Okay. So, what I want to key into during this portion of the presentation is the fact that the current COVID-19 strain shares an 89% nucleotide sequence identity to SARS coronavirus ZXC21, which belongs to the bats that we talked about. The bats that are living predominantly asymptomatically with this viral strain. And a 50% nucleotide identity to the MERS coronavirus. And finally, an 80 to 82% nucleotide sequence identity to SARS coronavirus, and hence why we name it SARS coronavirus 2, because it has the closest relation to the 2002-2003 epidemic outbreak. So, moving on with our discussion on the pathogenesis, or we're doing a snapshot now over human coronavirus, or coronaviruses as a whole, and their replicative process. Just a brief overture. And again, as I'm going to emphasize, I'm by no means a molecular geneticist or virologist expert, so all this is going to be kept pretty simple and going off of just the basics into how these viruses replicate themselves and to create disease in their hosts. Because it's my belief, a firm belief of mine, that the more we understand how these viruses create their progeny and continue to persist in their hosts, the more intervention can then be taken on trying to treat or eradicate the viruses altogether. And again, if I misspeak or I mention something that may have slight inaccuracies, I apologize. Again, I'm going off of resources that are on PubMed for the majority of what we know about coronaviruses, and I have all the resources included in my PowerPoint presentation. So let's get started with the snapshot of viral replication. And before we dive directly into the deets, as I put it here, for the replicative process for these viruses, we're going to talk a bit about the morphology. With the morphology, this is a discussion on the structural proteins that make up the virus and how it looks, as we see up in that image up there, and I'll have one come up on the PowerPoint slides as well. But it's a good idea to get a good image of how these viruses look, and more so the role that their surface proteins play during their attachment, entry, and subsequent replication. So, what we know about these coronaviruses is that they are positive sense. meaning as opposed to negative sense viruses that are RNA as well, the positive sense means they're most like our in regular cell processes in mRNA, messenger RNA, meaning as soon as it enters our cell, it is immediately ready to be translated into proteins that benefit its infectious process. So it's ready to go. It's geared up and ready to go. It doesn't have to make a subsequent copy of itself like the negative sense RNAs need to first when they enter the cell. So they have to go in the cell and then create a positive sense version of them for subsequent production of their viral proteins. So positive sense are just ready for action. And their single-stranded RNA. We talked about that for briefly earlier. Some unique features that go along with these viruses is an unusually large genome that I'm going to discuss again and again. It's on the order of 30,000 nucleotide-based sequences, or give a little, take a little. There's ranges out there from 28 to 32,000 kilobase pairs, or kilobase nucleotides, which is very large when you're talking about a RNA virus. And I put a little joke for humor in there to see who's paying attention to reading. No, the Corona does not apply to the beer Corona Extra. It's Corona extensions, which are little spikes that it uses to attach to its host for entry. And we'll talk more about that as we move along. So we already know all these informations we talked about. We keep referencing it. What else is there? What else do we need to find out? So there's 4 structural proteins that coronaviruses have, and they are the S protein for the spike, the M protein for the membrane, and the E protein, which is a very important protein we're going to talk a bit more about, and the N protein, which is the nucleocapsid protein that houses and keeps safe the genome that these viruses possess. And some, not all, coronaviruses have a 5th structural protein known as the HE protein, or the hemagglutinin esterase protein. And it's not known yet currently, or at least unto my knowledge, that the COVID-19 possesses such a additional 5th protein. But what it does in some other coronaviruses is it's a hemagglutinin esterase, so it's like splashing acid onto a host cell to aid better entry for the spike into the cell. So it's kind of just like eating away at the plasma membrane to aid for a much easier process of attachment, infusion, and entry, and subsequent replication. So coronaviruses, we talked about a little bit more, expanding on their very large genome. It's been subdivided or classified, however you want to phrase it, into 2 main divisions, such as, or that are as such. So we have 2/3. They found out 2/3 of these coronaviruses' genome encode for the structural proteins that we just talked about: S, M, N, and E. And about the remaining third, on the order of about 10,000 kilobases, is encoding for the non-structural proteins and accessory proteins, many of which are still under current research for exact pathogenicity and targets of therapy. And we're going to dive into those in subsequent slides. A little bit more esoteric knowledge for it is their helically symmetrical nucleocapsids, which is apparently uncommon for positive sense SS or single-stranded RNA viruses, and more commonly a characteristic of negative sense single-stranded RNA viruses, further differentiating the positive sense from the negative sense. and making this also another interesting feature that coronaviruses possess, having something that a negative sense virus has. So going forward, we're going to revisit the structural proteins again, like I said, the S, E, M, and N, adding more explanation to their function where relevant. But for right now, I want to just do a little bit of foreshadowing by talking a bit more on the E protein. So the E protein is very interesting, and I'll show you on the next slide where we have a beautiful image coming up of the beautiful or ugly, however you want to define it, of the coronavirus virion particle itself. And I'll show you that the E protein is the tiny little yellow dot, which is accurate since it is very small, about 8 to 12 kilodaltons, or kDa. It's very tiny. But interestingly, what makes the E protein so fascinating and a current target for interventions on therapy and more research is it functions both to, as we'll discuss soon, to assemble the progeny or the subsequent virions or virus-like particles when the replication process has reached its limit in the host cell, along with the M protein. They function together to make the assembly. And also, the E protein has it has an ion channel configuration, is what has been found. And with that, it has the ability to alter host cell secretory pathways, which is said to aid in the viron's release of the subsequent viron particles that have built up. And we're going to talk about that more coming up. So here we have it. And to briefly summarize the proteins, all these red blobs, the coronae or coronas, are the spike proteins that allow for the attachment and entry of host cells. And the two little orange dots side by side all over it, those are the M protein that help make the integrity of the structure of the virus capsid, in addition to the yellow dotted E proteins. So they function together to kind of make the spherical shape of it and keep the membrane or the nucleocapsid of the virus intact. Oh, human coronavirus snapshot of replication, the host cell entry and fusion. So this can be said to be the first step of the replicative process. You've got to get into the cell in order to achieve your replication. So these viruses do just that through the use of various host cell receptors. And there's been, so far, a few that have been identified, but of more importance, two of them that are linked to the SARS coronavirus and MERS. So SARS coronavirus and the human coronavirus NL63, and also COVID-19. We're going to discuss these receptors. So to back this all up, it may sound confusing. So for SARS coronavirus and human coronavirus, NL63, if you recall, is linked to causing one of the common colds. It's one of the 4 viruses that are out there in circulation, known to infect humans. They are both shown to utilize the angiotensin converting enzyme 2 receptor on host cells as their port of entry through the S protein association with that receptor. Whereas the MERS coronavirus, Middle East respiratory coronavirus, has been shown to use the receptor dipeptidyl peptidase 4 as their route of entry. But we're not here to discuss MERS, we're here to discuss coronavirus 2019, which is SARS-CoV2. Alright, and so something of note is that beta coronavirus grouping has been shown to have the ACE2 receptor predominance. Next, the S protein or spike, however you want to call that, Once bound to the host cell surface membrane via the ACE2, undergoes cleavage of itself into two domains, S1 and S2. No, these are not the heart sounds, S1 and S2, for those that are falling asleep on me. But that's essentially how they begin their entry process, by cleaving the S2. So here's the spike. Once it attaches to the receptor, it cleaves, and you have these two domains. That may not be exactly how it goes, but you get the idea. Now, fusion next happens, where after attachment and subsequent cleavage, the viral nucleocapsid membrane and the plasma membrane then begin to mix and fuse, and the virus sort of just goes right into the cell, to the cytoplasm inside a cell. And this process has been demonstrated to be mediated by the S2 domain, which I put in bold there, because since we know that the S2 domain, furthermore, what we know about the S2 domain amongst these viruses, amongst COVID and the previous SARS, is it's highly conserved in its sequence, the nucleotide sequence. So, meaning there's not much change in the genomics or the nucleotide sequence from virus to virus, which is a good thing when you're talking about certain preventive therapies later on down the line to prevent this whole process of fusion and entry to the cell to occur. So that's why that's bolded there. So next we're going to talk about translation. And translation is, when it comes to these viruses, I like to say it's where the money lies in terms of what makes them truly unique, is the process by which they accomplish this. Not just this, but then their replication as well. The whole process that coronaviruses exhibit during this, by which they replicate, is interesting and just under heavy investigation. And we're going to find out why that is. So, but for simplicity's sake, everyone can take a deep breath. I'm not going to go into the ins and outs and mention all the TLR sequences, the slipper sequence, or the pseudo-knot structures. Don't worry about that. I'll leave that for the virologists and the more in-tune clinicians that are currently actively researching this under a microscope. We're just taking a look at why this is so different and what we need to keep our minds open to as far as what to look out for in preventive measures and therapies. So we're just going to give you the big picture idea. Once inside the host cell, this is where the magic starts to happen. And I use this word magic here appropriately because it's still truly mysterious in nature, as we're talking about. As I've mentioned numerous times, it's unknown. There's a lot of unknown variables how this happens. But normally translation of mRNA sequences, they have these stop codons to regulate protein production by mRNA. And frame shifting does occur in normal cell types and eukaryotic cells, but it's typically abnormal when it's exhibited by this virus. The virus has become over time very proficient at using a frame shifting technique to translate its protein in a sequential process that's uninterrupted by stop codon sequences. So this aids both in the efficiency of it to create as many proteins as possible, making a suitable environment for ongoing replication within the cell, or subsequent true replication of the genome, and also may be said to function as an outpacing mechanism of our immune system. That's not been documented or said, but with reasoning minds we can deduce as much, because normally virus enters the cell, starts replicating, our immune system reacts because it's viruses that are slowed down because most of them don't have that frame shifting ability to go through the translation process to make all these pathogenic proteins as fast as possible. So, though that's not been proven, it's it's a strong consideration to be aware of. We're going to talk about a bit more about how exactly these viruses inhibit our immune systems to keep themselves alive longer and thus able to spread in our bodies, creating more diffuse infection. So we'll talk more about that in just a few slides. So now, finally, we have polyprotein cleavage and RTC assembly and finally the transcription. So it may seem, and I apologize if all of my slides thus far have seemed pretty wordy and everything. It's just, there's a lot of information to be had out there and not a whole lot of fascinating images, really. So I apologize. I have tried to add a little bit of humor here and there, but it's wordy. But anyways, back to the topic. Polyproteins, the virus that we're talking about, and we touched base already, produces various proteins in its genome, known as the NSPs, the non-structural proteins, accessory proteins, and structural proteins. What we didn't really talk about yet is the viral genome that has the true core genome. I'm going to talk about that again, and subgenomic domains. And it's the true core genome that codes for and is unique in the that codes for the very unique RTC, replicase transcriptase complex, another hot topic of current research interest. So, and it's from the subgenomic RNA, for which all of these non-structural and accessory and structural proteins come from, that create a lot of the pathogenicity exhibited by the virus. But the RTC, as we'll see, is truly unique in nature, and what is known to be only really the one notable product of the true genome of these viruses. So now I put in bold the formation of the certain NSPs via translation by and I failed to mention that the translation that's occurring is through use of our host cell ribosomes. That's how these viruses operate. They employ the use of our normal cells' ribosomes to translate. So, but anyways, this leads to the production of the certain NSPs leads to inhibition of the host innate immune system, and we're going to talk about a bit more in sequential slides. So, and as I already talked about, once a sufficient amount of subgenomic protein by the virus is translated by host ribosomes, which are an aggregate of polyprotein structures, the virus deploys various proteases to cleave these structures into non-structural protein accessory proteins. One such protease is PL-Pro or papain-like protease. Maybe a little bit of esoteric knowledge, but it's always fun to know new names. And anyways, the assembly of the RTC occurs through NSP association into the complex. So all those, the polyprotein structures, this massive production from the virus's subgenomic RNA gets cleaved. Yet again, there's cleavage occurring. And these proteins, some of the NS proteins, start to complex together to create the RTC, replicase transcriptase complex. And finally, the RNA transcription can occur once the RTC is assembled. But before it can begin the replication, another protein, yet another one, I know, but this one is important because it's assembled by non-structural protein 12, so it's one protein that has been numbered and named and we know of. At least that's the case for the previous coronaviruses, SARS-CoV. And it creates the RNA-dependent RNA polymerase. So RDRP, which is a component of some of the tests for, I bolded because it's one of the components that we use in the testings that are out there for the viruses. amongst other components as well. So, the unique transcription process by which the human coronaviruses replicate their RNA involves a sequence of events that is really just too complex and far-reaching for our purposes here. But I do want to just highlight what is unique about the whole replication process. Yet again, another unique feature of these viruses is that the ability it's how the recombination is allowed or manifested and used greatly by the virus through strand switching ability of the RDRP. So during replication, the RDRP can just hop from strand to strand and start the recombination process. And recombination, if some of you recall, is the use of taking other pathogenic factors from other viruses or quasi-species virus or RNA viruses out there and incorporating it into their own genome to aid and further virulence of the virulence and pathogenicity of that coronavirus. So it incorporates other traits of other viruses. So, and just a little tad bit more of information to go over is the unknowns for the coronavirus COVID-19 that are still of interest because they share no homology and are still largely unknown with COVID-19 is the open reading frame 3B protein and a product made by or encoded by ORF8 that the structure of that ORF8 encoded protein has no similarity with the other SARS coronavirus from 2002-2003. Okay. Next up, all right, assembly and release. So this is the last portion of the snapshot into viral replication of these coronaviruses. So once the replication nears its end for the virus within the host cell, the last step is, of course, packaging its genome back in and into a mature virion particle for subsequent release and infection of other cells, so spread within the host. Structural proteins are the last, so they're the first in, last out, if that makes sense to anybody. Meaning they needed to gain entry, and now, before we are ready to go and release this virus back into our circulation of our bodies to infect other cells, they've got to be translated again and assembled into the virion particles. So they orchestrate the whole assembling of the subsequent viral progeny, the VLPs or virions. And this all takes place once these proteins are translated. They get inserted into the endoplasmic reticulum of your host cells in our bodies, and from there they get transited to the ERGIC, which is the endoplasmic reticulum Golgi intermediate compartment, for assembly and association with the N protein, the nucleocapsid housing the genome protein. And an important step to note, which is of current therapeutic aim, as we talked about, and which is why the E protein was discussed at length earlier, was for this purpose here, is that even though the whole assembly process is sort of kind of orchestrated by the maestro protein, which is the M protein, maestro's appropriate use of that, and so it essentially takes all the pieces together to start forming the circular or virion capsule progeny. It needs to be associated with E. The E protein works together with M to create the structural integrity. And it's important to know this because in certain recombinant forms of coronaviruses studied, the viruses lacking in E protein in the surface of their nucleocapsid structure aren't always lethal. So it sort of defangs the virus, so to speak. So M plus E is needed to make me. That's just kind of my mnemonic that I've come up with to remembering this association. The N protein, although it houses the genome, so it's a very important protein, it doesn't add much in the sense of assembling all these proteins together. It just kind of is like an enhancement, kind of like a catalyst or a hurry up, but it just sits there and watches things go on and just says, all right, hurry up, let's go, and it may catalyze the process to move faster, but it has no structural purpose in the sense of the glycoprotein capsid. So the last protein to show up to the party is the S protein. And this is an intriguing little protein, as we've already seen before. But more so intriguing here is the fact that it's able to navigate through to the ERGIC by itself, solo. And then, if it misses its associated party there of the virion particles, meaning it's not able to be incorporated into the virions or VLPs, it then transits to the outer cell membrane of the host cell and starts to attach or attract itself to other neighboring cells and create pathogenicity that way, which interestingly is now is one step of how these viruses manage to evade host immune systems. So it's not known whether or not it's a mishap or whether it does it routinely yet, but it's just been shown to do that. So they can create giant multinucleated cells by having the spike protein go to the surface of your host cell, drags another cell together, and just over time get this mass multinucleated cell that is under the radar of our immune systems. So it can spread infection that way as well. Okay. And so for those keeping count, this, which I don't know if we've discussed it, but some of the NS proteins that have been produced by the virus are known to inhibit. So the non-structural proteins have blocking nature or blocking ability of our host immune system, the innate and the acquired. It's not known exactly which non-structural proteins do that. There's certain numbers thrown around out there, but again, a large majority of these non-structural proteins remain to be further researched. But what we do know is some of them do interact and inhibit the immune system via inducing apoptosis, which is programmed cell death of our immune system cells, further giving evidence to the fact that the lymphopenia seen in most patients is from inducing our white cells to die. So there's that interesting tidbit. Now, current unknowns still being investigated in COVID-19. Well, as we know, there are many current unknowns when it comes to this virus, a point with which we can all agree on after what we've just gone through. But to name a few important ones when it comes to its genomic identity, here they are. The exact mechanism of how the RTC works in replication is of high research importance and investigation because it's so peculiar. And how the virus only manages to package just the positive sense, full-length, single-stranded RNA out of the many different RNA species produced during infection. So how it has selective ability to take that genome and package itself again into the virion and release is still interesting and being researched. And again, as I've said before, majority of the non-structural proteins and accessory proteins remain unstudied. Okay. Alrighty. And what's also unknown is the presence of an intermediate host, which, as more information develops and we know more and more about this virus, and its ability to create infection in humans almost from the get-go, the discussion has now changed from even having, is there even an intermediate host out there? It could have just jumped to humans directly from the host that we've discovered this virus from already, which is bats and the civet cats. All right. And another unknown with the virus that's important to highlight, back it up here. Put it in there. Okay. So we already talked about how the multi-nucleated giant cells aid in this virus's ability to evade our host immune system. Another method of which it uses to keep itself from being undetected or blocking our host immune cells is the use of non-structural proteins that, interestingly, have been found to either block the signals for incoming host immune cells, the macrophages, which is part of the innate immune response, and also the acquired immune response for generating antibodies to neutralize the virus. Some of these non-structural proteins have been implemented in being able to deploy mechanisms to inhibit signals, or actually even promote signals that counteract or create apoptosis in the immune cells, killing them off, and that's why, as we'll see in the symptomology, common laboratory presentation amongst many patients is lymphopenia, plus or minus leukocytosis, which is just the increase in our neutrophil count.
So, Dr. Kogu, it's producing something that's killing the lymphocytes.
Yes.
Wow. Okay. Go ahead.
Yep. It's killing them or inactivating them or remaining undetected, but it is inducing apoptosis. And what I put in bold here, because it's very interesting how this virus interacts with NF-kappa beta, which is the main cell, one of the main cell signals in the immune response by all of our host immune cells, both innate and acquired. And it's very, very interesting how it's cell specific, meaning in our lungs it could use this signal to promote a pro-inflammatory response, which is aiding its pathogenicity, which is seeming it's counterproductive thinking, right? Because if you were going to promote a pro-inflammatory response, you're going to try and kill yourself. That's what but the virus in this case that. But in other host cell tissues,
because by getting that inflammatory response, it induces a cough, which helps it spread its virus.
Exactly. But also through the tissues itself too. It aids its cell-to-cell transmission. So it's using our host immune cells as a sort of superhighway, spreading through dendritic cells by amplification of this signal, amongst other signals. But in other cell types, it's been shown to shut it off completely. In, for instance, small intestinal cells and tissues. It's a very, very fascinating virus of how it's doing this, and it makes sense why it's becoming such a pandemic. It's using our own host cell immune system machinery against us and for itself. And this is just going to briefly discuss about, or we're talking now, pathogenicity and transmission. We've moved away from the replication, talking about the factors that, all the little intrinsic factors that aid this virus's ability to keep itself going and its progeny going. Now we're just going to discuss more about how that directly affects our cells in a virus-type infection. So, because these viruses have been shown extensively through the years in animal models who have extensive tissue tropism, the ability to infect certain tissues over others through its spike-like protein, and that's why we're going to talk about in a second here this whole discussion about the S and L type strains, whether or not they exist or not. But the S protein is a target of, as we talked about, research to find out more about how we can prevent entry and if there are more virulent strains out there. Okay, so
now you're saying that some people experience diarrhea with this as well as nausea.
yes,
Prior to the symptoms.
Yes.
So
Yes.
someone coming in and saying, "Oh, we got diarrhea," and everyone saying, "No, that's not a cause of that is incorrect."
exactly. And that's why at the whole start of the discussion, I talked about the grandfather strains of the PBEV and TGEV, the transmissible gastroenterovirus coronavirus strain, which is the more virulent form, coincidentally. And people at the CDC on their webinar talk on last Friday, Dr. Furukawa, or I'm sorry if I mispronounced his name, but when he was describing the symptomology, there were a number of patients coming in that developed subsequent more severe illness that presented on the onset with diarrhea, nausea, and vomiting in the gastrointestinal symptoms.
And same with, you said that some people present with just a runny nose.
Yes, and that's more, we're seeing the more common cold, quote-unquote, type symptoms of the rhinorrhea and upper respiratory infection symptoms in the population, the younger population patients, and more so in pediatrics. Though there's limited info on that exactly, but there is a large number of people presenting with minor symptoms.
Now, on your transmission, you talk about respiratory droplets
Yep.
versus fomites.
Uh-huh.
Now, is most of this being spread by respiratory droplets? Is that where the super spreaders are doing it? And
Yes.
and how much evidence do we have that fomites are a significant problem here, transmission?
To date, there is not much evidence to support the fomate transmission, but the CDC implements a common sense precaution and safety practice in healthcare settings and out there in the general public, meaning if there's high traffic sites, countertops in offices, door handles on entryways, all these things where there's a lot of people touching and coughing and leaning on surfaces, you're going to want to sanitize those before you touch and transmit it to yourself, because it's got to get to your mucosal membranes, and the hand is the best route to your face and where your mouth is and your nose, and it will go through your mucosal membranes, because the virus can live for several days on flat, non-porous surfaces, not exposed to UV radiation or excessive heat. So, and you want to use a 65% or 75% higher alcohol sanitation products. Everclear, I hear, works quite well too.
But probably not standard vodka, because standard
No.
vodka is only 40%.
40%, exactly. So, interestingly, there was a company out there, Tito, who makes Tito's vodka, that they're using their runoff from their distilleries, higher percent alcohols, and bottling it into disinfectants to give to people and sell for a cheaper price.
That's great.
So, okay. So,
Hopefully I can support him when this epidemic is over, or pandemic's over.
yeah, right. And what I put here in bold is the strong indication with the shortness of breath leading to more severe symptomology and the true presence of this virus. And that has to do with, because as we talked about, the extensive tissue tropism of these viruses and their selectivity for tissues, it seems to be affecting more of the lower respiratory tract. And all these symptoms that people complain about, the sore throat, rhinorrhea, seem to be as a result of lower respiratory tract infection.
Rhinorrhea, of course, means runny nose.
Yes, runny nose from the dry cough, coughing, and elevating our temperature as a downstream effect. But the primary pathogenicity lies in the lower respiratory tract with COVID-19.
Now, a lot of people have talked about viral load.
Yeah.
How is that really a factor? Or is it? One wonders if a super spreader can infect a large group of people in a meeting in, say, Boston, if you need that high of a viral load, or if it can be or a low viral load that's transmissible.
As you know, in the slide to come, we're going to talk exactly about that.
I will let you talk about it.
So, we're just going to wrap this one up with the laboratories we just talked about that they also shared on the live webinar stream of this, by the CDC on the 13th, 2020, March 13th. And what they are describing is, again, the lymphopenia, frozen lymphocyte, or very low lymphocyte count, and either leukocytosis or leukopenia, and now more and more discovery of AST and ALT enzymes being elevated, which also points to GI involvement. So, interestingly though, procalcitonin levels as amongst a number of other pro-inflammatory markers have been shown to be normal on the onset of disease, with slight elevations and variations throughout the clinical course, which the clinical course is still being tacked down. There's a median of 21 days post-symptom onset that people are still symptomatic and shedding RNA virus. And we'll talk about here shortly, the shedding of the RNA virus in stool, urine, etc. It's still unknown whether or not this is a significant risk for viral transmission. So what we do know so far is that it's respiratory droplets through the air transmission is what that means. And person to person, you have to be within 6 feet, which is 2 meters, and not wearing any personal protective equipment and stuff like that. And so we just talked about was that even people who have symptom resolved, 1 to 4 weeks, they've been shown to shed the RNA virus. But again, it's still unknown whether or not it's a significant risk factor for ongoing transmission. And what's important to note is COVID-19 patients are cleared of isolation upon 2 consecutive negative RNA tests collected 24 hours apart. That's when you can lift the isolation protocol and instruct them to, still remain isolated, but in your own home and don't go out into the...
So after you're asymptomatic, you can still be shedding virus, and so you need 2 separate tests that are negative by over 24 hours.
Correct.
I mean, just emphasizing what you said.
Yes.
And so this is how, I think one case in maybe Italy, he tested negative and then went out and potentially spread it to Italy.
Mm-hmm.
And but he had a negative test. And the greater than 24 hours, how good is that? And do they need to be testing other than nasal and throat? Should we doing rectal swabs? What should be tested?
Dr. Barry, you're hitting all my points. Actually, are the topics of interest. That is exactly correct. Though there's limited evidence, and the CDC hasn't come out and outright said to be testing routinely the stool and urine samples, though the published article on PubMed in February this year is advocating for people who are presenting with the GI initial symptomology of gastroenteritis that they should be candidates for, if they have a negative screening from nasopharyngeal swab on subsequent respiratory development of symptoms, that they should then collect stool and or urine samples to be tested for these patients, as we'll talk about. That's highlighted in those sequential slides. Although CDC hasn't come, to my knowledge yet, they haven't announced doing that routinely.
Okay.
So everyone knows.
So I guess the question is, and it's going to be one that I think a few people want to know is this something we can pick up on toilet seats, or using the bathroom, that someone else, when they use the bathroom, becomes particulate in the air and gets spread that way in a public toilet?
Well, I would hope, people going to use toilets in public restrooms routinely have basic sense to do hand hygiene, but as we've discovered, a lot of people surprisingly don't practice good hand hygiene. So it is theoretically possible, sure, that you can pick it up from toilet seats, because if you can get it from foamite on surfaces that are highly trafficked, like countertops and flat non-porous surfaces, why should a toilet seat be no different if a respiratory droplet has landed on it?
But again, there's not been any proof of
There's
that.
there's no proof of that, but it has to come in contact with mucosal membrane. So if someone who is going to the bathroom, wiped, and then didn't wash their hands,
Okay.
there may be some transmission to occur there. Contact with mucosal surfaces from fomite.
Got it.
You know, because then they use their hand too and go and touch their face and mouth. Because on average, I don't know how accurate this statistic is, but the average human being touches their face something around 23 times per hour, or roughly 552 times a day. That's what the CDC posted a long time ago, and other sources have put their own numbers in there too. But anyways, we like to touch our face a lot, subconsciously, going to bed all the time. So, and I don't know if you've tried or anyone at home has tried, but I challenge people, see how long you can go without touching your face. Suddenly, everything starts to itch. It's very difficult. You know, it's hard to do.
Maybe pour Tabasco sauce on your hand.
Or jalapenos.
Yeah, rubbing jalapenos, and then each time you touch your face, you're going to be like, oh.
Yeah. Shouldn't have done that. Yeah. All right. So, everyone knows by now. If you don't know, then you should remove yourself from out from underneath the rock. But 2 to 14 days, with recently a 2 to 9 day post-exposure from being exposed to someone who's symptomatic to you developing symptoms is the incubation and onset of symptom time period. So, with a median time period about around 5.1 days for people developing symptoms after contracting the virus. And again, going over the symptomology, we'll go in more detail with, but it's a gradual onset of the fever, which is not always present in cases, especially in pediatric populations, as we'll talk about. But then the subsequent development of myalgias, malaise, the fatigue, and soreness, and cough. And right now, studies have shown that around the second week or after 8 days of symptoms, it either maintains the clinical course of being mild, or it can now subsequently progress to more severe symptomology, onset of the shortness of breath, with a median onset or average onset of pneumonia, pneumonitis around 9 days of infection.
If we're tracking the onset of this virus amongst the population, and we institute a strict quarantine, it should take, what, 5 to 9 days before we start seeing a curve bend, if it's gonna bend?
By the numbers, yes, of around about 5 days, that would be correct. So you isolate them, yeah.
But as late as 9 days.
As late as, well, no, that's the 9-day factor is more in reference to people who go on to develop more severe infection. So that's for people who have had severe infection. They started developing that at the turn around 8-9 day transition. They got the pneumonia, pneumonitis, ARDS, etc., and we'll talk about those going forward.
So 5 days plus whatever testing time there
Correct.
is.
Yeah.
So in the state of Washington, it's been up to 144 hours
Yep.
by one lab and 72 hours
Oh yeah.
by another before at least we get the results here.
And we're going to drive home that testing point near the end of this presentation too, which is not far from now. So, but we're going to talk about all those details. So, again, yes, this is from the CDC webinar 3/13/2020, which is this past Friday. The severe disease is noted by the progression of as follows: pneumonia, ARDS, sepsis, septic shock, multi-organ dysfunction, and multi-organ failure being the end of the line, so to speak, or if they do recover, which is hard to recover from extensive multi-organ failure. And I included this snapshot because I think it beautifully illustrates, it's hard to see on there, but It's from ScienceDirect.com, and it is another article that came out more recently, March 3rd of 2020, and they use the data from Chinese public health to make this illustration of symptomology.
So it's worth going over.
It is.
Go ahead. So you have fever,
Yes.
cough, fatigue, increased urine production, headache.
Yes.
Headache. Any meningeal signs that you know of?
None that have been reported. No photophobia or neck stiffness, any stuff like that. So,
Runny nose. Go ahead.
So it's divided into systemic disorders or symptoms and respiratory disorder symptoms. And on the screen's right-hand side, but left up here for me looking at it, we've got the fever, cough, fatigue, sputum production, headache, hemoptysis possibility, acute cardiac injury, because again, tissue tropism for the viruses, it's been shown to directly affect cardiac cells.
So how would that show? Is it a heart attack or just elevated troponins? Or
I would believe elevated troponins and enzymes, but no sources really come out outright and said that, so I'd have to double check that source, but it's just been shown to cause acute cardiac injury. My bet would be CKD. Hypoxemia, dyspnea, lymphopenia, diarrhea. Again, and I wish I could circle that for people to see, because it's... so anyways, then the respiratory symptoms, or respiratory isolated disorders, is a runny nose, sneezing, sore throat, pneumonia, ground glass opacities, RNA anemia, which is just elevated viral count in the bloodstream, acute respiratory distress syndrome. And to circle back to the ground glass opacities, and I believe I put a note in here. We're talking about the superstars now, but just to quickly, briefly explain the ground glass opacity findings, that yes, more and more people are showing that who have severe disease onset, but the CDC has come out and said on Friday not to do chest X-ray or CTs on the outset of diagnosing, because early findings is on those imaging modalities is normal. It's only once they develop more severe shortness of breath, dyspnea.
That you get ground glass opacities.
Correct. Yes.
But you can get ground glass opacities from other viral.
Yeah. So RSV. Yep.
RSV. Okay.
Exactly.
So it's not pathognomonic. It doesn't necessarily equal it, but it's a strong suspicion. If everything else ruled out that you may have COVID-19.
It's strong suspicion, and also a indicator of more severe disease onset. Okay. So, but as going forward, CDC advocates against the use of X-ray and extensive imaging for diagnostic purposes in the beginning of symptoms.
Okay.
Because you're right, RSV, respiratory syncytial virus, has been known to cause it. Many viruses can cause the ground glass opacity. So, all right. Now we got the topic of what is a super spreader. So, as Dr. Barry, you mentioned, they are, and I've kind of equated this to a dose-dependent analogy, meaning the greater the dose, the greater the spread, and the greater the viral load. So all that stuff goes hand in hand. You got to have greater inoculation. So what this means is super spreaders have a viral infection that shows enhanced viral replicative ability, and though it is not known what causes this still, but as we just reviewed extensively before with the replication and everything going on there, it can be any number of those factors that's causing this, any portion. And though it was thought that this was due to the, quote, L-type more violent strain that they found in China in the epicenter, Wuhan City, they sampled 103 cases. They were finding all the similarities of the more violent cases had this L-type abnormality, or basically what it was is the S spike protein encodes for anywhere between 621 to 689 nucleotide sequences. And the sequence that they were finding these changes that were similar across the 103 cases were just about 1 to 2 sequence or nucleic amino acid changes. That's it. So 1 to 2. And further investigation going forward has found that those sequence changes had nothing to really do with the pathogenicity of the virus or the S protein's function to bind and go into host cells. So
it's an interesting aside. I've had friends in different places, and at first, whenever this virus appears, there seems to be certain myths that occur. The first one is, don't worry, it's already throughout the entire community, we're already immune to it.
Yeah.
And then events seem to show that to be false.
Yeah.
The second one is, oh, we have the less virulent strain.
Correct. Yeah.
And then subsequent events show that to be false.
Exactly.
And then the crisis hits after everybody minimizes and ignores it and says, why not? I have a friend who was here, then went back to Mexico, and she's saying the same. She's hearing the exact same things in Mexico, in the exact same sequence she heard here 3-4 weeks earlier. So it's an interesting
and
self-delusion.
Exactly, and what is, not to get too political with it and everything, but what is aiding in that is the misinformation spread. So the media outlet is coming out with all these things and linking people to all these different little theories and downplaying this in some instances and then upplaying in others. So we have all this information with wide swings that it's just leading to a mass misinformation spread. And what I'm trying, what I hope to do at the end of this presentation is to narrow that down and keep it to the facts and the simplicity. And as we already know, these viruses are still to this day highly researched because of their unknownness. So much is unknown about them, and they create disease and outbreaks that are pandemic potential. And as right here I've highlighted, the reason why 2002, 2003 SARS outbreak back then was easily, or not easily, but compared to now, much easier contained from spreading, was due to the fact that there wasn't as many super spreaders in that outbreak that they've come out and said in the research articles. So, just to recap, a super spreader, someone has some portion of this virus, some type of sequence is encoding for the virus to replicate at a heightened ability and just go and go and go and just produce this massive viral load and just basically pushing on it, just for layman's terms or illustrative purposes. It's like poking the Pillsbury Doughboy. They're going to release a huge spray of the viral load in the air. So as people that have a higher viral replication and production, and there's more of these people with this current COVID-19 outbreak out there with super spread, that the R0, which is the reproductive number, is greater than the 2.3 to 3.8 ranking that has been determined for this virus, which is similar to the SARS back then. As an average, as the whole numbers go, 2.2 to 3.8 is, so for every one person infected, they spread it to approximately 3.8.
And sort of like the HIV initial infection, you get a very high viral load before you become symptomatic or with initial mild symptoms, and that's when you're most likely to be a super spreader. Is that correct? That you get a higher viral load pre-symptomatically? So
information is still lacking on when along the disease course people are truly transmissible. So we're going by right now symptomology onset as the greatest risk for transmission.
Okay.
Though that could be certainly very true. Someone who has the super spreader ability of the virus can transmit the virus without even having any symptoms. That is definitely a possibility. But as of right now, someone's sick, coughing, you see them on the street, you want a wide margin of greater than 6 feet at least to bypass them. So, sick contacts. But the R0 for super spreaders has been difficult to track down. It could be much greater though from what certain statisticians.
That we're hearing.
Oh yeah, so much greater than 2.2 to 3.8. That is the average to also the SARS and MERS back then that we've already seen, those strains. The R naught here, though, could be hundreds. One person spreading to hundreds. Or thousands, correct, yeah. So, that wraps that up. Now we got this, and I put that little effect in there of swiveling around. We've already preluded to it. The S and L strains, is there concern? Is there a difference? Largely, since the time that the information came out, no. It's not been proven that there is a true S and L type based on a 1 to 2 nucleotide deletion or change. Because it has been shown to not affect the pathogenicity of the virus. And as we see today, with the ongoing massive spread, if that were true, it wouldn't have gotten to this point so far. Okay, so a couple more points to take home from this is well, actually, that wraps that up. Yeah, we covered it, so it's going to be available for those who want to read more about it. But we talked about it.
Basically, it's a myth.
It's basically a myth, debunked. Though, is there a more virulent strain out there? Sure. Do we know what denominates it yet? No. So, what I highlighted here, UFC 2019. No, not the Ultimate Fighting Championships, but the Ultimate Flu versus Coronavirus comparison. I don't know why it does it all the time. I want this yellow thing to come up last, but whatever. So anyways, flu. What we know, sudden onset. People who are infected with influenza viruses, typically, the onset's very rapid. They wake up in the morning, just bam. You got hit by a semi-truck, aches, pains, fever, cough. Doesn't have to be all in one day, but certainly it's faster than the coronavirus, as we'll talk about. Kills thousands of people each year, approximately 35,000 cases in the U.S. alone. Millions of people worldwide affected every year. Mortality rate of roughly 0.1% annually, and that's been a statistic shared by CDC and other public health industries out there, or not industries, but institutions out there for the past 10 years, with about a 0.005 or 0.05% fluctuation year to year. But if you apply that to the numbers, and I've written it down there, it's, well, we'll circle back to that once. I want to first bring up the coronavirus in comparison. So, COVID-19, it's a gradual onset, as we talked about for symptomology. You get the either low fever or no fever, gradually progressing to aches and pains, cough, rhinorrhea, the runny nose, which are not always present again. Or it could start from the GI. You get the GI symptoms, diarrhea, nausea, and then you finally have the respiratory symptoms. It's a stepwise, gradual progression. The death toll, rounding, and I updated this actually last night, over 6,000 cases globally. What was it, just a week ago, we were in your office talking about this, and it was 4,000. So it's concerning. The potential for billions to be infected, as we see right now with all these the crises. Right now, we've got the governor, he issued all restaurants and bars last night to be closed.
Yeah.
Yep. So
unclear whether he's closed the Indian casinos yet. And
unclear.
This weekend they were packed. And,
yep.
From what people were telling me, and one super spreader in India casino could wipe out all the ICU capacity in the state.
Yeah, that is, and that's another hot topic of the CDC and everywhere else is the conserving the healthcare capacity model right now.
Mm-hmm.
We'll talk about that in the end, Chris.
It was interesting that he had exempted them.
Yeah.
But, all right.
All right, so what else do we know? Mortality rate fluctuating from 2.7 to 3.4%, but just recently it's proving much higher than that right now in certain populations, and they're taking now the days since symptom onset in certain cities and populations, and applying that to the modality. And in China, for instance, they've reanalyzed the numbers and found actually a 20% mortality rate given a certain population.
Wow.
So that's been just the other day released.
Wow. 20%
So,
mortality.
among certain populations, when they compared the numbers, when they actually reanalyzed things, yes, from the onset, days of symptomology to death.
Wow.
Yeah. So I have to update that, but I was hesitant because I wanted to still wait for more information to develop. But as time goes on, we're seeing this become more and more true.
Wow.
And worrisome is no vaccine, although trials are underway. And we're going to talk more about the treatment in the end because there's more information added on that as far as last-ditch efforts go and hopeful up-and-comers.
And no baseline human immunity to anything like this.
No, but I'm going to talk about that in this. We're going to highlight this immunity in the sense of people who have resolved infection, using those and going back to the old school methodology of taking the resolved patients' antibodies and giving that to more severely infected patients as a hopes of neutralizing the virus. And that's been proposed by the professor of immunology from Johns Hopkins University, Arturo Casavidal. He said that could be ready as soon as a few weeks. And also, it's been shared that the Dutch, quote unquote, were the first to develop antibody therapy right now. Mm-hmm. Whether or not that's true, it needs to be further investigated.
It'd be shocking if the Chinese didn't. They had the
right,
and they certainly have a large population.
Correct.
That's
Yeah.
gotten over it.
That is shocking. You know, so there's hopes for antibodies to be given to the more severely affected patients to neutralize infection and slow the rate and development of ongoing viral pathogenicity. So, okay, the virus is not intended for the human system, and that's highlighted here again. The reason why it's such a huge issue, our bodies have never seen this, so we don't have the immediate immune response to pick it up, and that's via intrinsic methods to immune cells that we're not going to go over and cover here. It's not an immunology course right now that we're going to go and discuss all the ins and outs of that, but long story short, we don't have the recognition of these viral pathogens, so it's free to spread, and also further dampen our immune system should start showing signs of active fighting back. It's already using our signals, and we talked about, of our host cells against us, so it keeps itself blocked and free to spread, and actually taking some of our cells and making it worse against us. So, very fascinating, very interesting, but very frightening virus. So, let's see, pediatrics population, OBGYN. I had forgotten to include this information, but I did, never fear. I wrote down the few title topics. They're not that long of what's been known because there isn't much information as of yet to be shared, but the CDC on their webinar did cover this, and what the CDC has said is based off of still, again, China, their Wuhan population of infected patients. So pregnant patients who are affected, of which they only had a small case to use, is around 34 cases that we're looking at, with a median age of around 30 years old, all within the second to third trimester, with a median post-conceptual weeks of 36. And infection started for a large majority 2 days before delivery, with a range of 13 days pre-delivery and up to 3 days post-delivery. That's the range. 13 days before you deliver, up to 3 days after is when all of them develop infection. And all the procedures for delivery were C-section, and subsequent detection of COVID in the offspring has all tested negative thus far of those patients. So further aiding, and also on analysis of the amniotic fluid, cord blood, breast milk, and placental tissue after being subsequently tested and retested, all of those tissues were negative for the COVID virus infection. But right now, again, as we talked about, it's too small of a case to go off of. But the CDC is using that.
So in these cases where they developed it both before delivery and after delivery, there was no virus in any of the newborns, any of the amniotic fluid, cord blood, or any of that. And did the mothers do well, or how did they do? Was there any risk of?
Yeah, we're going to talk about that. But just to answer what you're saying, is since the majority of the delivery methods, not majority, they were all C-section. We don't know if there is a vaginal route of transmission, but based off of the subsequent testing of tissues in that region and beyond, all the other stuff that we discussed, cord blood, breast milk, which breast milk is aiding the fact that these patients can still continue to breastfeed but exhibit hand hygiene, which we're going to talk about, the CDC covered. But there's nothing supporting thus far a placental transmission of the virus.
Interesting. Okay.
So, could there be vaginal? Because we talked about mucosal surfaces. Sure, that's a possibility, but nothing has been extensively come forward about more information. We still don't know enough yet. But yes, pregnant women are more susceptible to infection given the physiologic changes of pregnancy, and the CDC also is mentioning this as well. They have increased oxygen consumption, heart rate, and a shift away from cell-mediated immunity during pregnancy. That's the normal physiologic changes. So all of that makes them much more susceptible to respiratory pathogens. COVID being no different, obviously. Now, of those patients we talked about, the small case that the CDC is using as their information source from China is that only 2 of the mothers were subsequently admitted to ICU care for critical care going forward, and of the 2, one of them delivered a viable pregnancy and the other one delivered stillborn. And testing has not been available of that stillborn whether or not he had infection, so we don't know.
Okay.
But only 2 of the 34 have been labeled severely ill. But yes, pregnant women are more susceptible to infection.
Not that 2 out of 34 is not an insignificant number.
Exactly. Yes. Not an insignificant number when you look at it as a ratio of that small of a caseload. Sure.
Statistically indeterminate.
Yes.
Because it's such a low number.
Yeah.
Okay.
But as far as the CDC's recommendations for how we should handle pregnant patients in the U.S. right now is if it's a pregnant woman is suspected or confirmed COVID-19 positive, or a PUI, person under investigation, and she's on her way to the hospital, proper contact by the patient or family member should be made to the hospital so the hospital can set up proper precautionary protocols, making sure that have the proper PPE available to handle the patient incoming and subsequently putting them in an isolated room, which is much more difficult when we're talking about obstetric wards. Some clinics are not retrofitted for that.
Well, I could see where intubating the patient and doing a C-section would be beneficial for the child, no exposure, and also beneficial for the healthcare workers because of the closed circuit on the ventilators that they use.
Yep.
As where a woman in delivery is going to be hyperventilating and spreading virus.
Oh yeah, super spreader. There you go.
Yeah.
So, furthermore, a separation. So, according to CDC, transmission after birth via contact with droplets from respiratory secretions by the mother is a concern. Hence, necessitating the or basically HCP, healthcare personnel should be in touch with the mother and discuss with the healthcare team the potential for mother and baby isolation separation protocol.
So cognizant of that, and weighing that against the bonding issues of separating them.
Exactly. Yes, and
that's a tough one.
Yep, and thus far, until transmission-based precautions are discontinued, which again could be the 14-day range or longer, or depending on if the mother is suspected or if she's truly infected. So, and risks and benefits of temporary separation should be discussed with mother by the healthcare team. Separate isolation rooms for infants while they remain PUIs, because infants, if they're born to a mother with confirmed COVID and there is the risk of transmission, though not been confirmed yet, you're still going to want to talk to the mother of the possibility of the separation protocol until the baby's been tested negative and mother seems to be managed through covering the face with a mask, etc. So then that plays into what the CDC has said. If rooming in or co-location occurs via the mother's wishes, she still has authoritative. I don't know if that's the case now, as this gets worse and worse, but or it's unavoidable is being the more likely scenario due to the capacity of healthcare being overwhelmed or on the verge of being overwhelmed. The rooming in is becoming more and more the consideration going forward. So if that is to take care, engineering control should be employed, so that would encompass, they say, to use the physical barriers of a curtain between mother and baby in the same room, and keeping that distance at greater than 6 feet to decrease the rate or the transmission rate from the respiratory droplets. And the breastfeeding should be encouraged, although they do advocate for the use of a pump by the mother if she's symptomatic and is confirmed COVID positive, with subsequent sanitization of the breast area and nipple and the pump, the parts that and the pump that comes in contact. Sanitation before and after each pumping. And if the mother truly wishes to have breastfeeding, then she should be instructed and watched and supervised before the breastfeeding occurs, of that she's got the proper hygiene down, and she exhibits that each time before feedings. And she has a face mask over. So it can be still performed, though it's obviously a greater risk if she's symptomatic.
So the face masks decrease spreading from the infected person to the public.
Mm-hmm.
Not as much preventing the public from getting it.
Correct.
Standard face mask.
And we're gonna summarize that and highlight that later too, but that is correct. Yes, the face masks need to stop being bought in bulk by people that are concerned out there, because the reality is, mask or not, they are just as susceptible out in public areas as the average person not wearing a mask.
They're actually making it worse because the people who should use it are
Yep.
the people who are infected, and they should be able to, who has a cold or symptoms, should
Yep.
be wearing a
Exactly.
face mask. Maybe a diaper too.
Yeah, maybe, as we found out, yeah. Diapers as well. Okay, and one last fact, and that's going to be on the pediatric population. Even more limited information when it comes to pediatric populations, again, based off the China model. They found that 2% of the infected population during the outbreak was children or younger people under 19 years of age, with a fluctuation to certain smaller city and communities of 15% of the infected being younger adults. As
It made me think,
yeah,
I wonder if passing gas is a cause of super spreading. Weird thought. Go ahead.
weird thought, but plausible, sure. Different form of respiratory droplet. Anyways.
Or using the urinal, because there's some splatter on urinals.
Exactly. Yeah.
And,
Sure. Splash it on your hands. You. Yeah.
It also splashes into the air.
Into the what?
You get some splashback on urinals.
Yeah.
Wondering if that's.
It could be. What we didn't talk about the super spreaders very briefly is the CDC also mentioned as much, is not just their ability for the high viral load and subsequent high aerosolized particle production. But of those particles, which usually drop to the ground and dissipate, they're finding with super spreader people who are more infected, these particles are staying and lingering in the air around the patient for longer. So the ability of the virus to still stay suspended in the air increases the likelihood of someone else walking into the room or by them and not having the PPE appropriately on, or say the patient was in this corner of the room, was coughing, they moved him 6 feet over here, but he was coughing over there, and now the healthcare worker comes in where he was over there, not had protection on, and gets the transmission that way.
There you go.
So you've got to be careful with having the PPE before entering any symptomatic patient's room. So that's what the CDC has announced, that the ability for them to have the virus lingering in air is a concern.
So more HEPA filters are needed.
Exactly. HEPA filters, definitely. So just to wrap up with the pediatrics, before we go forward, to summarize and complete this topic, again, largely unknown, as we're finding out from a lot of these things. It's still to be determined. But from what they've seen with the younger populations being affected, the majority of them, and which makes it very difficult in the sense of being able to stop the spread, is the majority of asymptomatic people and people with minimal symptoms are the younger groups, which is why closing down the schools was a good thought and good implication right now, but should have done much earlier, is because now you're sending them home and they could be asymptomatic but transmitting to their family, and the family could go out and get supplies and continue the spread. So anyways, that aside, it's much difficult to contain the virus spread.
Spilt milk under the dam.
Exactly.
Or under the bridge.
And so and also because the co-detection rate, which was shared on Facebook by you, Dr. Bauer, with the ID conference on March 8th, in which they talked about the co-detection rate being 2%, meaning if you test positive for another virus, it's that virus, no need to test for COVID. When it comes to the pediatric population, unfortunately, and still more information and research is needed, co-detection rate where the virus is higher.
Oh, interesting.
So yes, so RSV, flu, mycoplasma, that's on CDC. It is.
So the kids can have all of it.
They can, and so that can lead to misrepresentation of the test results.
Also means that if this virus is picking up stuff from other viruses, it's the kids that will most likely produce the most wicked and virulent strain.
Correct. Yep. Most definitely.
All right.
So that wraps up that when it comes to pediatrics. And of all the cases, there was only one of a documented case that was, I don't know that age exactly, so I'm not going to mention it until I look at it, but he did have subsequent critical illness, and that was only one out of all the cases that they've shared so far.
Okay.
And whether or not he had underlying disease is also unknown. And also CDC announced on their webinar that there's misrepresentation of the information previously that's still on the CDC website, that underlying illnesses, for pediatric populations is still unknown if it's a concern. They came out on a webinar and said, yes, it is. So if you are a pediatric or a younger person with underlying health issues of note, high hypertension, we're talking 160s over 100s and stuff like that, or high swings. So hypertension, diabetes, cardiovascular disorders, HOCM, hyperobstructive cardiomyopathy, anything like that, if you've got a pediatric population, that is a risk for more severe illness as well, just like adults.
Asthma or previous lung injury, or
that hasn't been really investigated much, the asthma, but as far as like lung injury, like emphysema from alpha-1 antitrypsin deficiency disorder.
how about like preemies?
Oh, so bronchopulmonary dysplasia from the high oxygen.
Yeah.
That also, no information has come out about that. And I was actually asked about that recently by a family member. No resources has brought forth that information, but it would stand to reason if it's a significant amount of damage, you're more susceptible. Less lung tissue that's functional is going to put more strain on the functional parts, and you have more severe illness.
Okay.
So, but we'll await what the CDC says going forward, but that would be the theory right now.
Yeah.
So that's for the pediatric population, OBGYN. Any questions?
As if I hold them back.
Yeah. Prevention. So, cannot stress this enough. Isolation, isolation, isolation. And I'm going to just take a brief rant here, is because on Sunday the other day I was driving back from the hospital here. The amount of people that I saw, because the sun's out and people were out on the roads driving, was, in this current crisis and situation, disheartening. Really disheartening. People going out with sunglasses, walking around. I saw people jogging and running like not a care in the world. Sure, you should exercise and stuff like that, but the lack of prevention protocols being exhibited by people.
But how much is transmitted outside?
It's still to be determined. But, with the recent change in our climate, being colder, arid climates, and the wind that we have in Washington, if there's been a person in a populated area that's a super spreader, it has the lingering particles, and you've got a gust of wind coming in, and there's 10 people over here, maybe greater than the 6 feet. It's not been confirmed, but I'm just using basic geometric progressions here and models like that.
I'm gonna say I want my patients out exercising.
Yeah.
I think cardiovascular disease still just kills more people daily.
Yeah.
Than this, although that may change briefly. And I think I would rather have them outside exercising. What disturbs me was going past the bars and the grocery stores and seeing those packed this weekend.
Yeah.
And
that's, yeah.
You know.
That's what I'm more getting to. I'm just saying, people are certainly exercise should be promoted, but if you can, try and do it indoors. Especially not around public.
But not at a gym.
Not at a gym. Indoors in your own home if you can. You can get it. How many American families have treadmills? There's quite a significant amount.
I would still rather have them outside exercising.
Okay.
I'm
in remote locations.
No, I'd still rather have them outside on the sidewalk or their house walking.
Okay.
I'm an exercise nut, so that's my prejudice.
All right.
And maybe I'll be proven wrong.
Yeah.
Well,
regardless, but that's definitely
most importantly, I don't want people breaking the habit of exercising.
Yeah.
Because long term, that will have a much more deleterious effect.
No, I agree.
So
yeah.
I'm telling people, go out and exercise.
I agree.
I'm pushing back on your statement, but go ahead.
What I think I'm failing to mention is, sure, outside and stuff is great, but not outside in crowded areas.
Oh, yeah.
So that's what I'm talking about. Like parks, if there's a lot of people at the park in one area, if you've got a super spreader out there, you run through their super spread mist, you've got infection.
Yeah.
So, yes, outside.
I wouldn't go to an outdoor concert.
Exactly. There you go. No, the large gatherings
where
avoidable.
people are close together. Yes.
Correct. So that's okay. All right.
On an unwindy day.
Yes. And indoors, obviously, if you've got to go, everyone has to shop. It's unavoidable. But if you're going to go in, this might be the certain scenario in which a face mask could benefit you if there's a large amount of people and you have to get supplies. That maybe, but you don't need 1,000 masks for that purpose.
Well, again, has that been shown that it prevents you from getting it or just giving it?
If there's a large amount of people shopping at the same time and some of them are sick, I would say a mask is a good preventive mechanism in that situation. I'm not saying to go out and buy them wholesale and stuff, but
okay,
one per family.
Is there data that shows that the face mask prevents you from getting it?
No, but in the hospital setting, people symptomatic, you place it on them to prevent the spread, and healthcare personnel protect themselves. But I would assume it's not been stated, but that model could be placed to very crowded shopping facilities.
So people should wear it on themselves to prevent giving it to other people.
Yeah.
All right.
Yeah.
I'll agree with you
Yeah.
on that one.
Yeah, you know, that's true, because you're not going to go out there wearing goggles and the whole face shield to truly stop yourself from getting it in the store, so a mask wouldn't really help you much in that sense.
Yeah.
So, that's correct. Anyways, so isolation, though, is key. It's the fastest way to drop the R0 number, and recently a lot of epidemiologists and statisticians have come out, and everyone by now has seen that. I didn't include it in the slides, but that whole flatten the curve model.
Mm-hmm.
You got the spike, here's the healthcare level, the spike of people that are coming in, getting tested, or that are at risk and getting infected as what we want to avoid, and the capacity of people infected right now that we can take care of. So flattening the curve, best way to do that, isolate yourselves. Isolate yourselves and proper hygiene, wash your hands, everyone should do that. I kept the inclusion of the Vietnam Public Health Administration video because it is fun to watch. I want people to go watch it. It is fun. And sanitizing of like we talked about, flat, non-porous surfaces. The hard one, the high fomite trafficked areas. So door handles, doorknobs. Also, you should wash your hands always, or sanitize your hands before touching your face. Cannot stress that enough. And masks, we talked about already, so I'm not going to go over it again. Long story short, let the masks be utilized in the healthcare setting, symptomatic people, and prevention to spread to healthcare workers. Respirators, we touched bases on. Well, we will. We'll talk about the respirators, but very briefly. Everyone knows about the N95 respirator. And here we go. Isolate. But we're gonna take a pause on that. But talking about, this is more in reference to what hospitals have been doing, whether they've had major outbreaks, like the San Jose hospital that was shared with by you, Dr. Bauer, on Facebook. The findings of what they started implementing in their hospital setting was, and CDC on the webinar on Friday has said something similar, is when you've got patients of undifferentiated pneumonia or respiratory illness, taking those patients, placing them in contact and droplet isolation for 2 to 3 days, or in a separate unit of the hospital, sort of like a quarantine precaution, and watching for subsequent development of symptoms to pin the diagnosis on COVID or the resolution from another viral illness. And CDC also said if there's been COVID-19 positive patients, to cohort them and isolate them in their own unit together as well, for being able to maintain capacity of the healthcare system. And current recommendations going forward for healthcare personnel is to implement and use the alternative methods for visiting patients who are ill, and that's the use of telemedicine, the car visits for testing. You drive up, it's like a drive-through swab and go, and phone consultation hotlines. This has all been updated recently and verified again on the 13th by the CDC. And the CDC.
Now, I've also heard that now Medicare, since Friday, since the presidential order was signed, will now pay for telemedicine visits on par with regular office visits. So healthcare providers should be switching to telemedicine in this juncture.
Mm-hmm.
Go ahead.
Okay. All right, and the exposure risks and protocols, all found on CDC. So I didn't go into much in depth about this because it's easy to find information, and there's plenty of scenarios that are case specific. So where here I've included the general considerations and common sense reasoning. Obviously, the greater the exposure, and exposure is measured by time and distance. So duration of how long you're going to be in the presence of the symptomatic patient and the distance from that patient in that room. Those are your greatest indicators for your risk of exposure. And of course, that astronomically increases if it's a super spreader that you're in the room with, which we have no way of finding out whether that is. How do we do that? As of current testing methods right now. So, time and distance is the best way to assess your level of exposure. And anyone who, I think they've changed it. Now all levels of exposure, they recommend a respirator use just because of how far it's gotten. But medium and high risk, we're going to stick to as being the first use, especially with this disruption in supply chain, which is the biggest point of discussion of the webinar on Friday, was to conserve and reuse certain important pieces of medical equipment, the PAPRs, the face shields, stuff like that. And what they're saying is the respirators, after doing testing on models, that they can be used beyond the manufacturer date if necessary, because they still have filtration of air been shown beyond the date of expiration. But the issue, it's made to grade, so other components of the respirator or mask are falling apart, like these straps to keep things, the face fits. That's why the expiration date and shelf life is timed.
The plasticizers start to wear out.
Exactly, yes.
Rubber bands and stuff.
So, but they'd say, to use the ones that have expired in shelf lives to use this for training purposes and fittings, not necessarily for direct patient interaction. And the use of face masks are, as per the CDC, acceptable to use in patients who are intubated that have appropriate hookups for the ventilation in the negative air ventilation rooms. But what the respirators that are in limited supply in the hospitals right now should be conserved and used for until supply resumes are patients who are to undergo aerosolized inducing procedures, so the BiPAP ventilations before the hookup of the filtration, and people who are any other procedure that's going to develop that. So there's numerous ones that develop more aerosolized particles, and that's what the respirators should be conserved for. Okay, and of the three forms for healthcare personnel to monitor the onset of symptoms, there's three main ones: self-monitoring, active monitoring, but now currently I've updated and CDC is recommended against is the third form of self-monitoring, which involves two modalities, both the healthcare worker's own personal facility and public health. They basically, the previous recommendation was that it was mandatory for them to go and get tested by both institutions to show for clearance of symptoms. That's no longer necessary if they're feeling better.
They have to, the whole system's overwhelmed.
Yeah, exactly. And there's a finite number of tests, which we're going to talk about. But self-monitoring means the healthcare personnel should monitor themselves for fever by taking their temperature 2 times a day, and monitoring for any progression of other symptoms: sore throat, shortness of breath. So that's that. That'll be available for people to read. And here's the link to the CDC. I told you the table of risks, exposures, recommendations for what type of personal protective equipment to wear. And we're not going to go into that. There we go. Testing, big topic of discussion, as we already alluded to. 2 main modalities being employed are the PCR and microarray sequencing, which are just methods of testing that amplify and sequence the RNA genome for greater imaging. It's like Photoshop, taking a picture, chopping up, or enlarging it, and depixelating it. That's how I equate this to explaining how these tests essentially. But the most general terminology. Anyways, and though there's a lot of numbers being thrown out there about the sensitivity, specificity ratings of these tests, it's not enough to definitively come out and say what those true numbers are.
We're sort of flying blind with these tests.
Yeah, exactly. Flying blind, and as we see, that's what the whole point of this presentation was, to show the complexity of the viral process and the proteins that are made, and there's only a certain number of them banked. It's easy to miss certain components if it's a variant that's mutated before testing or whatnot, or if it's not in that tissue that you gained a sample of, because of the immense tissue tropism exhibited by the virus.
Tropism meaning?
The ability to affect other tissues. So, it could be in your gut rather than your lungs, but you have lung symptoms as well. It hasn't just fully moved there quite yet. So, and biggest point for testing is, and I bolded it there, and again, it's not been outright said yet, but I anticipate it will be soon, the obtaining the stool and urine samples for testing in people who test negative but have all the symptomology markers of disease. And that, more importantly, at the onset of symptomology, presented with the GI disturbances, diarrhea, nausea, vomiting, or one or the other. It doesn't have to all be three of them. Okay, and another important portion of the test is 4 to 8 degrees Celsius when obtaining the sample. It should be placed in that temperature rating and placed on dry ice or ice pack to be sent testing overnight. Now, if there's a delay expected, greater than 72 hours, the sample should be placed in negative 70 degrees, 70 degrees Celsius liquid nitrogen to be frozen and sent accordingly to be tested. So, as we can see, not just because of the complexity of these tests involved and all the different intricate steps involved, and also the limited number of facilities still offering tests, surprisingly, with the universities coming on board. Although Johns Hopkins announced the other day that they are able to produce test results within 3 hours, but they have the 24-hour buffer of when they can truly get results back. But they say as little as 3-hour turnaround. So that's something to keep an eye on, their ability to do testing and have rapid turnarounds. So the need to get more universities on board with the testing is important. But right now, limited resources, limited testing kits is an issue, in addition to all the other mishaps in the testing process. Treatment.
Where are the testing kits coming from? Are they coming from China?
That is a good question.
Okay, continue on.
So, okay. From my understanding, I do not believe, I think the CDC has made their own testing kits as well.
Okay.
It's on the CDC website.
Okay.
I apologize. But each university right now are making, actually no, I do know this question. The Chinese Public Health had released the viral genome sequence to be used for the testing.
I'm talking more about the swabs.
For the swabs? Oh, okay.
The media. Are those coming from China or from the U.S.?
I know that the universities that right now are coming on board, they have their own that they're using. But for CDC as using the test kits, I have to confirm that.
All right. It'll be interesting to see if that supply chain is available
Right.
also. Go ahead.
So treatment, there is currently none, although there are treatments being investigated. And I'm sure everyone's seen by now remdesivir in the compassionate care use for severe illness patients. Through the manufacturer, they're allowing that. Chloroquine is also another anti-malaria medication being investigated for therapy, amongst other antivirals being used that we've already had on the market with varied results. But remdesivir is the star child of medications going forward.
And where does that work on the virus?
So that inhibits the RNA polymerase, so it disrupts the ability for the virus to replicate itself and undergo its progeny.
All right.
So it's a nucleotide analog antiviral.
And it works on this coronavirus RNA polymerase? nice. That
Yes,
we know
so it
does.
stops, it encodes for a disruption in the replicative process early on in the replication, so it can attenuate the virus essentially.
Okay.
Just like it does for other viruses in HIV. So it's a nucleoside analog.
Okay.
So, or nucleoside antiviral nucleotide analog use.
Well, it's interesting that. All right. Go ahead.
And also we talked about the possibility of using antibodies from patients with resolved infections to be deployed right here, right now, in a few weeks' time to give to severe patients of severe illness. As mentioned by immunologist Professor Arturo Casafadal from Johns Hopkins University. Okay, generally speaking, comes viruses. Supportive care is the name of the game, or vaccination. But since we have no vaccination right now, the investigation to all those therapies we just talked about is of utmost importance as this virus continues to spread and create more cases. But I'd like to key in on kind of easing tensions too during this, is that the facts are right now 80% majority are going to have a mild clinical course when they get infected. An even higher percent of people who get infected still do end up surviving. But in terms of sheer numbers, the mortality rate of this virus is concerning, but it's not the end of humanity. So people should just be instructed to isolate themselves, hand wash, basic hygiene. So, and wash for symptoms. Okay. And this is the fact versus fiction stuff, which we can skip over. We've already done that. People want access to seeing they can test their knowledge for it. They can have these slides later. Okay, but that about does it.
Thank you, Dr. Coley.
Mm-hmm. Any questions?
No.
Okay.
We answered it.
Okay. Essentially, but the most general terminology. Anyways, and though there's a lot of numbers being thrown out there about the sensitivity specificity ratings of these tests, it's not enough to definitively come out and say what those true numbers are.
We're sort of flying blind with these tests.
Yeah, exactly. Flying blind, and as we've seen, that's what the whole point of this presentation was, to show the complexity of the viral process and the proteins that are made. And there's only a certain number of them banked. It's easy to miss certain components if it's a variant that's mutated before testing or whatnot, or if it's not in that tissue that you gained a sample of, because of the immense tissue tropism exhibited by the virus.
Tropism meaning?
The ability to affect other tissues. So, it could be in your gut rather than your lungs, but you have lung symptoms as well. It hasn't just fully moved there quite yet. So, and biggest point for testing is, and I bolded it there, and again, it's not been outright said yet, but I anticipate it will be soon, the obtaining the stool and urine samples for testing in people who test negative but have all the symptomology markers of disease. And that, more importantly, at the onset of symptomology, presented with the GI disturbances, diarrhea, nausea, vomiting, or one or the other. It doesn't have to all be three of them. Okay, and another important portion of the test is 4 to 8 degrees Celsius when obtaining the sample. It should be placed in that temperature rating and placed on dry ice or ice pack to be sent testing overnight. Now, if there's a delay expected, greater than 72 hours, the sample should be placed in negative 70 degrees, 70 degrees Celsius liquid nitrogen to be frozen and sent accordingly to be tested. So, as we can see, not just because of the complexity of these tests involved and all the different intricate steps involved, and also the limited number of facilities still offering tests, surprisingly, with the universities coming on board. Although Johns Hopkins announced the other day that they are able to produce test results within 3 hours, but they have the 24-hour buffer of when they can truly get results back. But they say as little as 3-hour turnaround. So that's something to keep an eye on, their ability to do testing and have rapid turnarounds. So the need to get more universities on board with the testing is important. But right now, limited resources, limited testing kits is an issue, in addition to all the other mishaps in the testing process. Treatment.
Where are the testing kits coming from? Are they coming from China?
That is a good question.
Okay, continue on.
So, okay, from my understanding, I do not believe, I think the CDC has made their own testing kits as well.
Okay.
It's on the CDC website.
Okay.
I apologize. But each university right now are making, so, okay, actually no, I do know this question. The Chinese Public Health had released the viral genome sequence to be used for the testing.
I'm talking more about the swabs.
For the swabs? Oh, okay.
The media. Are those coming from China or from the U.S.?
I know that the universities that right now are coming on board, they have their own that they're using. But for CDC, as using the test kits, I have to confirm that.
All right. It'll be interesting to see if that supply chain is involved, is available
Right.
also. Go ahead.
So treatment, there is currently none, although there are treatments being investigated. And I'm sure everyone's seen by now remdesivir in the compassionate care use for severe illness patients. Through the manufacturer, they're allowing that. Chloroquine is also another anti-malaria medication being investigated for therapy, amongst other antivirals being used that we've already had on the market with varied results. But remdesivir is the star child of medications going forward.
And where does that work on the virus?
So that inhibits the RNA polymerase, so it disrupts the ability for the virus to replicate itself and undergo its progeny.
All right.
So it's a nucleotide analog antiviral.
And it works on this coronavirus RNA polymerase?
Yes, so it stops, it encodes for a disruption in the replicative process early on in the replication, so it can attenuate the virus essentially.
Okay.
Just like it does for other viruses in HIV. So it's a nucleoside analog.
Okay.
So, or nucleoside antiviral nucleotide analog use.
Well, it's interesting that, all right, go ahead.
And also we talked about the possibility of using antibodies from patients with resolved infections to be deployed right here, right now, in a few weeks' time to give to severe patients of severe illness. As mentioned by immunologist Professor Arturo Casafadal from Johns Hopkins University. Okay, generally speaking, comes viruses, supportive care is the name of the game, or vaccination. But since we have no vaccination right now, the investigation to all those therapies we just talked about is of utmost importance as this virus continues to spread and create more cases. But I'd like to key in on kind of easing tensions too during this, is that the facts are right now 80% majority are going to have a mild clinical course when they get infected, and even a higher percent of people who get infected still do end up surviving. But in terms of sheer numbers, the mortality rate of this virus is concerning, but it's not the end of humanity. So people should just be instructed to isolate themselves, hand wash, basic hygiene, so, and wash for symptoms. Okay, and this is the fact versus fiction stuff, which we can skip over. We've already done that. If people want access to seeing they can test their knowledge for it, they can have these slides later. Okay, but that about does it.
Thank you, Dr. Coley.
Any questions?
No.
In closing, I would like to just give some recent updates upon further research that I did for Dr. Bauer here in some things that may have been a gray area or left to question. But upon further review, and as per the CDC updated guidelines in the public health laboratory testing for COVID-19, they have updated and announced in their literature, and I quote, "As of the evening of March 15th, 84 state and local public health laboratories in 50 states and the District of Columbia have successfully verified COVID-19 diagnostic tests and are offering testing. This includes one or more public health laboratory plus DC and Guam." And to address the other issue that might have caught some people off guard or raised some back of the neck hair there when I was mentioning the mortality rate being as high as 20%, where I got that statistic. I didn't just pull it out of thin air or materialize it. So to give people a reference, it's through thelancet.com and just above the graph, and I'll go as quoting it right now. Here we go. Quotation: "A recent time delay adjusted estimation indicates that mortality rate of COVID-19 could be as high as 20% in Wuhan, the epicenter of the outbreak. These findings show that the current figures might underestimate the potential threat of COVID-19 in symptomatic patients." So that's just highlighting the very severity of this that I sense is still being handled as not as grave. And so I hope that after watching this video, people gaining more of an understanding into the inner workings of the virus, we can start coming together as a society and as a people to combat this. And the best way is isolation until relevant therapy is out in the market. All right. Thank you for listening and watching.